Okay, everybody, so this is where we left off with the chapter 16 PowerPoint. We left off with talking about the different types of white blood cells. So make sure you know that the technical term for white blood cells is leukocytes. And there's several different types of leukocytes. They can kind of be put into two different categories. First, we have the granulocytes. They get that name because they look like they have little granules inside of them, like little kind of bits of grit or dirt inside of them. So they're called the granulocytes. And this includes the neutrophils, the basophils, and the eosinophils. So you can just remember all the granulocytes, they end in phils. Neutrophils are the major type of granulocyte. They're also probably the most important white blood cell to the non-specific immunity. They are highly phagocytic, which means that they perform phagocytosis, which we're going to talk about in a second. But you guys probably already have a good idea about what phagocytosis is. That's when a white blood cell encounters a pathogen or a microbe and essentially kind of grabs onto it and ingests it. And that's how they kill them. They kill them using their lysosomes. Basophils have a role in inflammation and in allergies because they release a cytokine called histamine, which I'm sure you have all heard of antihistamines before. So histamine is one of the major cytokines that are involved in allergic reactions. And then eosinophils are involved in defense against parasites and parasitic worms. So oftentimes in the United States, you would not find, people that have very many eosinophils because we don't have a lot of parasitic type infections. But in other countries, certainly you would find a lot more eosinophils in people's blood. Then we have the agranulocytes. So agranulocytes are the ones that don't have, the grit kind of thing inside of them. The first type are called monocytes, and monocytes become macrophages. And macrophages are also extremely important to the nonspecific system in the same way that neutrophils are very important because of how phagocytic they are. So macrophages are by far the best white blood cell at phagocytosis. We also have dendritic cells, which are found mostly in the skin and mucous membranes, which also perform phagocytosis in kind of a non-specific type way. And then we have lymphocytes. So please do not get the term lymphocyte and leukocyte confused because it happens all the time. Leukocyte is the term for all the different white blood cells. Lymphocytes are one specific group or class of white blood cell. We have natural killer cells, which we'll talk about here in a second. They are part of the non-specific system. And then the T cells and B cells are part of the specific immune system. So we're going to hold off and we're going to talk about T cells and B cells in chapter 17. So you definitely want to know the job of all of these cells. I can almost guarantee you that every single one of these will have a question on the test. If you want to look in your book on page 447, there's a really neat picture of kind of what they all look like, and it kind of gives you an idea for their sizes and that kind of stuff. Now, I'm not going to test you on knowing what each and every white blood cell looks like, but it's really interesting. And then especially for any of you that are in the histotechnology program, you'll definitely need to be able to tell the difference between all the different leukocytes. All right, so natural killer cells, again, a very specific type of lymphocyte, and you can probably guess from their name what they do, okay? They are very good at killing things, specifically killing pathogens or microorganisms. So just like most white blood cells, when they encounter a foreign cell, they're going to release all kinds of different types of proteins, different types of cytokines, as well as some special proteins. One protein is called perforin, which causes pores to form in the plasma membrane of the target cell. and granzyme, which actually enters the cell and causes the cell to self-destruct, essentially, very similar to the process of apoptosis. So I kind of say, like, if some white blood cells are kind of like the police and some white blood cells are kind of like the army, the natural killer cells would be like the highly trained, specialized, like assassins, like the highly trained Navy SEALs of the immune system, because they are so good at killing these bad guys. And the thing is, they're pretty rare in the body. Like, we don't have a whole lot of natural killer cells, and we don't know a whole lot about them in terms of how they function and everything. So it's definitely a big area of research in immunology. If we can get, if we can kind of harness these natural killer cells a lot better, we can certainly utilize them to our advantage. All right, so phagocytosis is the process in which our white blood cells engulf and digest foreign cells or material. The macrophages are the best at this, right? You can even see the word like phage in their name. So macrophages develop from monocytes, and they're constantly patrolling the body for bacteria to destroy by phagocytosis. Now, there are some pathogens that we know of that are highly, highly resistant to phagocytosis. And here's a list of them. And, some of these we've already talked about. So Streptococcus bacteria, so, Streptococcus pyogenes, Streptococcus pneumoniae, highly resistant to phagocytosis, so very difficult for our macrophages and neutrophils to kill that type of bacteria. Same kind of thing with Listeria, which we haven't talked about Listeria yet, but we will. Certain types of Staphylococcus bacteria can resist phagocytosis. The HIV virus is very good at resisting phagocytosis. Mycobacterium, which is the cause of tuberculosis. And actually, when we talk about tuberculosis, we're going to talk about how they basically use phagocytosis against our own body. So not only is tuberculosis not able to be killed by phagocytosis, they actually use it to their advantage. Plasmodium, if you remember way back when in lab, it was lab 4, when you looked at eukaryotic parasitic type organisms, Plasmodium is what causes malaria, and Trypanosoma is what causes African sleeping sickness. So those two parasites, which are protozoans, they are also very resistant to phagocytosis. So you can see here, some pretty serious diseases. We've got pneumonia, we've got strep throat, we've got MRSA, we've got HIV/AIDS, we've got tuberculosis, malaria, African sleeping sickness. These are some very, very serious, very dangerous, deadly diseases. And one of the main reasons they are so deadly is because they resist phagocytosis. They can't be killed in the classic regular way that our immune system knows how to kill things. So the collective term for all white blood cells that perform phagocytosis is phagocyte. So really what we're talking about when we say phagocytes, we're talking about macrophages, neutrophils, and dendritic cells. Those are really the main ones. So here is the big bad picture in your book. This is on page 451, and this is very, very important for you to read over, look over, make sure you understand all the steps, make sure you know what's happening. The reason that this is so important is because this will be a short answer question on exam 3. So I will ask you the question, describe for me the steps of phagocytosis, and you have to describe these steps that are here, okay? Now, you don't have to write every last single thing word for word, but certainly, you need to demonstrate to me that you have a good, full understanding of phagocytosis from beginning to end, okay? So again, the picture's on page 451. It's the exact same picture that I have up here. So you need to explain to me, what is chemotaxis, what is adherence? Ingestion is kind of obvious. The macrophage would ingest the foreign cell. And then you have the formation of what's called the phagosome, which is a vesicle that has the bacteria inside of it. And then the lysosome, which you learned about lysosomes in AMP1, the lysosome is going to fuse with the phagosome, and the lysosome is what contains the digestive enzymes. So the foreign cell, the bacterial cell, it's usually a bacteria, the bacterial cell will then be digested by the enzymes that are found in the lysosome, and then any other like residual material, any kind of indigestible, polysaccharides or whatever, they just get discharged through the process of exocytosis. So, that is phagocytosis kind of as a whole. So again, just make sure you can kind of describe these steps, make sure you define the terms like chemotaxis and adherence, phagosome, digestion, you want to make sure you're defining things. You don't want to just give me a list of terms. All right, so inflammation, a cascading effect in the body. Now, obviously, it can be from an injury, it can be from like a broken leg or when you fall down or something like that. But what we're really talking about, of course, is microbial infection. So we're talking about inflammation from an infection. The signs of inflammation are always the same: redness, pain, heat, and swelling. So it doesn't matter whether it's a bacterial infection, a viral infection, if you fall down and hurt yourself, if you burn yourself with a chemical or something like that, inflammation is always going to be redness, pain, heat, and swelling. Now, normally when we're talking about an infection or an injury, it's going to be acute inflammation. When we're talking about chronic inflammation, that could be something from, a more serious injury. It can be from, like an autoimmune disease or some other kind of metabolic disorder. Usually a microbial infection is not going to really cause chronic inflammation unless it's a very chronic, long-lasting disease. One good example would probably be something like Lyme disease. Lyme disease is actually more of a chronic condition than an acute condition, and it certainly will lead to inflammation for sure. So the function of inflammation is to actually destroy or remove whatever is causing the problem, and then eventually repairing the damaged tissue. The only problem is if inflammation gets really, really out of control, it can do a lot more damage than it's actually, helping. So why does inflammation happen, and why is it always the same for signs? Well, because it's linked to this vasodilation. So vasodilation is the opening up of the blood vessels, okay? So basically, anytime that you have an infection or an injury of any sort, your brain really only knows one way to react, and that is to open up the blood vessels. So the brain says, okay, well, if we're injured or if we're infected by a bacteria, we're going to need blood cells to go to that area because we're going to need to fight off any sort of infection or any sort of problem that may be going on there. So the only way to get a whole bunch of white blood cells to that area is to open up all the blood vessels. So the opening up of the blood vessels is going to cause the release of various different types of cytokines: histamine, kinin, prostaglandin, leukotriene. All of those cytokines are going to cause an even greater increase in vasodilation. So this is actually, inflammation is actually an example of a positive feedback mechanism. So vasodilation is going to cause further vasodilation because of this release of these cytokines. And then also it's going to attract the phagocytes, okay? So the brain realizes that there's a problem. Let's say, it could be your leg, it could be your arm, it could be your liver, it could be your kidney, whatever. The brain says, okay, there's a problem, there's an injury or an infection, we need to deal with this, so we're going to open up the blood vessels. The blood vessels open up, that causes the release of these cytokines, that attracts things like macrophages, neutrophils, that kind of stuff, but then it's also going to further increase vasodilation. So if you increase localized, blood flow, which is what's happening, okay, the blood is going to flow into that area. It's going to cause the area to warm up because the blood contains things that make it warmer, okay? It's going to cause redness because the blood is red. It's going to cause swelling because that tissue is not meant to have so much blood flooding into it, okay? And then the swelling is what leads to the pain. So usually it starts off as redness, and then warmth, and then swelling, and then pain, because the swelling up of the tissue presses up against the pain receptors, and that's where you get the pain. So we already talked about phagocytosis. That's the next stage. So the first stage of inflammation is vasodilation. The second stage is phagocytosis, and then finally tissue repair. So anything that's damaged or dead has to be, flushed out and removed. That usually happens through the lymphatic system. And then healthy new tissue has to be put down, through the process of mitosis. So mitosis is that kind of healing effect, in the area. So, inflammation always has those four signs, because we're really just talking about vasodilation, and then phagocytosis, and then finally you have to heal or repair. So fever, we all know what fever is, okay? So that's when your body temperature goes above 98.6 degrees Fahrenheit. And the hypothalamus, you should have learned a lot about the hypothalamus in A&P1. The hypothalamus does lots and lots of things in the body. It's a very small part of the brain that has lots and lots of jobs. And one job that it has is it maintains your body temperature. It's basically your thermostat. It's your body's thermostat. So when we have an infection, especially if it's an infection with gram-negative bacteria, we already talked about the gram-negative bacteria, the endotoxin, how that creates a high fever. Well, what happens is as gram-negative bacteria gets released, as they get destroyed, as they get killed, that causes the body to release interleukin-1. Interleukin-1 is a cytokine that causes the release of hypothalamic prostaglandin, and that is what triggers the increase in body temperature, because your body knows that the bacteria does not like it to be much hotter than, say, about 100 degrees. So if your body turns up your temperature to 100 degrees or 100.3 or 100.4, what your body is trying to do is kill or cook the bacteria away. That hotter temperature is going to prevent the bacteria from being able to grow and multiply. So, in a lot of ways, a moderate fever is a good thing. It's an important part of trying to keep you from getting sick. Now, of course, I'm not a medical doctor, so you don't have to listen to me. You can make your own opinion about your own body. You can make your own opinion about, about this. But, if you're a relatively healthy adult, you're not a baby, you're not someone who's elderly, you're not someone who has HIV or cancer or who's had an organ transplant, right? So you're just regular, normal, healthy adult with a healthy immune system, right? A lot of times, a fever for just like maybe one day or 48 hours can really do a lot of good, because what can happen, and you don't want to run to the cabinet and start chowing down on the aspirin and the Tylenol and all that kind of stuff. Just let your body kind of do its thing. If you wake up and you're not feeling so good, you take your temperature and it's like, 99.9 or it's like 100.1. Then you can say, all right, let me take off work, let me take off school, let me get, lots of extra rest, drink plenty of water, and like support your body to get better. And a lot of times you end up, not being sick for as long, and you end up better for it, but if you wake up and you're like, oh no, it's 100 degrees, let me take all this Tylenol and just go to work and get over it, you're probably doing, more harm than good. Now, if you wake up and your temperature is 103, then yeah, you want to definitely do something about that. I mean, that's a much more kind of dramatic, out of control kind of fever. But just read up on some of the information about fever and how it can actually be beneficial type of thing. But it depends on the situation, of course. Okay, so some more antimicrobial substances that have a non-specific effect on, immunity or just keeping the body from getting sick. The first thing is called the complement system or complements. Now, I don't spend a lot of time on the complement pathways. They're really, really specific and they're very complicated. So what I want to do is I just want to kind of tell you a little bit about it, kind of define it for you a little bit, and then I'll just have you refer to the book. You can read more about it in the textbook. There'll probably be like just one question about in the test. It's not going to be a huge deal, but certainly if you want to read about it in the book, I think it'll be good. So the complement system involves the liver. So essentially, the liver produces some very helpful proteins that can help destroy microorganisms in the body. So it's kind of like the liver's contribution to the non-specific immunity. There's 3 different pathways that exist, okay? You have the classical pathway, the alternative pathway, and the lectin pathway. It's all described in your book on pages 456, 457, 458, and 459, okay? So you've got about like 3 or 4 pages in there that you could just kind of read up a little bit about it and just so you know a little more about the complement system, but it's not going to be a big part of the test at all. Interferon, we've already talked about. So interferon are antiviral proteins that can help in a viral infection. And the really interesting kind of neat thing about interferon is interferon proteins are species specific, not virus specific. So it's just simply called human interferon. You don't have different interferons for like the flu or the common cold or coronavirus. It's literally these proteins that are specific to humans. So like there's dog interferon and cat interferon and human interferon. But the cool thing is if someone is having, a really, really bad case of a viral infection and their body is not producing enough interferon to support the immunity, you can actually give them, it's almost kind of like a blood transfusion, but it's an interferon transfusion. It's almost, it's kind of sort of similar to giving someone antibodies. So you can actually synthesize these interferons and give them to someone as kind of a medication, which is sort of interesting. And then finally, we have transferrin. Transferrin is an iron-binding protein that helps to prevent microbial growth. So if you remember, back in chapter 15, I told you how much the bacteria love iron, right? They want to try to steal iron from us, and that's one of the main ways that they make us sick is by taking our iron away. So this is kind of like our answer to that, right? So the bacteria have the siderophores, which allow them to kind of steal away our iron, and then our bodies have transferrin. Transferrin binds the iron to try to keep it away from the bacteria. So the bacteria is trying to steal our iron with the siderophores, and we're trying to keep it away from them with the transferrin. So it's kind of like that battle back and forth over the iron. All right, and that is it for the rest of chapter 16.